all about it once class starts. Look at y'all just waving away. And we will open this one. Everybody have a good weekend?
I
I can use a paper towel if you want. Drew, help Jordan out. I know. So Caitlin's brother, who remembers James? Yes, a lot of people remember James. James moved to college this week. How was it?
I didn't move him in because I had rehearsal.
Okay.
And so, my mom said she only cried when she was leaving.
That's what she
And
said.
I don't believe her.
I told her she did better than me. Yeah. When we dropped Cole off at Baylor, I cried a little bit, but I lost it when Drew hugged him. I could have made it through all the rest of it, but Drew hugging him threw me over the edge. I like that. Hey, that is a motto. Does everybody see what the back of her hoodie says? Can we all live by this?
It's one of the rules when I go out with friends and my mom will present. She's like, "What are the things?" And the first one is don't act a fool.
Don't act a fool.
We act fool,
You act fool?
by the way. But I don't like overly act fool.
Okay, well let's not overly act fool. I'm gonna drop. Now we have this is a fun day. We started with heat and now we're switching to air. You know, it's that time of year. All right, cool. All right, I'll let Jordan finish nuking lunch and then we will go ahead and start. Anybody else do anything fun this weekend? What'd you do? Anything?
I didn't do anything, but I learned something. I saw a headline somewhere that some scientists are thinking that the sun is conscious. Have you heard of that?
No, I have not heard of it.
That's like a concept of an analog horror.
Okay, that's interesting. Yeah.
Like
I learned that we're living in a simulation.
Oh, we're living in a simulation?
Yeah.
Does that mean I can stop paying taxes and everything?
Yeah.
Because I'm in if that's the rule. Instead, no.
The sun used to look yellow instead of white.
Oh, I think that's atmosphere, but okay.
And
Yeah.
time's going faster too. No.
Oh, okay.
That's enough.
Oh, Coyote had the flu?
He is not contagious anymore, but now he's back at work.
Oh, good. Well, you did a nice job taking care of him, I'm sure. Yes,
It's
Lucas.
actually like a 50-50 chance whether we're in a simulation.
Okay.
So
50-50.
So let's if you can simulate worlds, let's say like we don't know if we're in a simulation or not. So if you take it's possible that there's like one top world and then a ton of simulated worlds, like simulations in simulations. If you take all of those worlds, including simulation ones, and throw a dart, are you likely to hit the top one, the real one? You're not. But because we don't know we can simulate worlds, it's 50-50.
Top one happened to be the real one. Why can't it be the middle one?
No, because that's the real world that's simulating all the others.
Yeah.
Okay. Stop conversation. Let's get to our topic before we scare away our guests. All right. Y'all are such a fun group. All right, so we are talking about protists and fungi today. So let's see what we've got. Your homework assignment talked about something we touched on in class, a prion called bovine spongiform encephalopathy, right? Or BSE, mad cow disease. So then you had to do more about it when you got home. So in 1986, something strange began to happen to cattle in Great Britain. Some were acting strangely, losing control of their movements, and eventually dying. Farmers watched helplessly as the disease they called mad cow spread through their cattle. The disease affected more than 30,000 cattle in 1991. The moms and I in this room probably remember this. This was fun times, right? British scientists carefully researched the veterinary histories of the 169 sick cattle, and all 169 had been given feed enriched with meat and bone meal protein from slaughtered cattle. Okay, we are forcing cows to be cannibals, which is kind of bizarre and it's weird. And yeah, it's not what they do, right? What are cows supposed to eat? Yeah, they're herbivores. They're not supposed to be eaten, but we would giving them more protein because we were trying to, I guess, make the beef more protein rich. And Didn't work so well for us, did it? Could this practice spread a disease? The meat and bone meal substances added to the cattle feed had been sterilized at high temperatures in excess of 100 degrees Celsius during processing. How could any infectious agent withstand that? Well, we know now prions can withstand that, right? Scientific researchers studied the brains of the cattle killed by mad cow disease, and they found that large areas of the animal's brains had been destroyed. Under the microscope, it resembled a sponge, right? And because of that, it's bovine spongiform encephalopathy, or BSE. And it was a mystery, and as you read about it, you learned a little bit more. So remind me, what is a prion? We talked about it last week. Is it a bacteria? Is it a virus? Is it something different? What is it? Ridley?
Mutated protein.
Yeah, it's a protein. It's not a virus. It's not a bacteria. Which of those two things is alive?
Bacteria.
Are viruses alive?
No.
Correct. Very good. So it's a protein and it becomes denatured. It folds itself incorrectly. And we've talked about this before. You got a chain of amino acids. It makes a protein. Once it's folded into a particular shape, it's like, which origami shape do we need to do this job? And once it unfolds, it cannot function and do that job anymore. So we had this protein getting into the nervous tissue, particularly the brains of certain organisms, and just basically destroying it, right? All right, so when and where was BSE first diagnosed? Who remembers? Do you remember?
1986.
Okay, very good. Yeah, 1986 in the UK, and you answered all these online, so this is just what do you remember? How many BSE infected cows have been found in the US, and when and where did they occur? Claire?
3 were found. One was found in 2003. Another one was found in 2004.
Mhm.
And then I think the last one was found in 2005.
Yeah, something around that. I think that's about right.
I think what we said there was 13 cows.
So in all, there were 6 cases, 2 in Alabama, 1 in California, 1 in Florida, 1 in Texas, 1 in Washington, roughly 2003. Don't think we've had any since the very early 2000s because we have learned.
It said the last case was 2023.
Oh, was it?
Yeah.
Oh, I missed that. Okay, there was a new one then. Oh, I forgot that. All right, so there is a new one. You're right. So we know better, but still, and some of these, like we talked before, some of these prions, like in particular kuru. Do you remember the incubation period for kuru? 10 to 50 years. Yeah, so you don't know you have it most of your life. So what country stopped importing beef from the US? How about it, Caitlin? Mm-hmm. Yeah. Anybody else remember any of the others? Do you remember? Oh, Eunice?
South Korea and Mexico.
South Korea, Mexico. Yeah. So basically, in 2003, after we said, oh, we have some tainted cows here, most countries were going to ban it, including Japan, South Korea, Mexico, and Canada, which together purchased 90% of our beef. Ooh, that's a hit, isn't it? Yeah, that's a hit. So how much did Creekstone, which was this organization that you read about in conjunction with this, contend it lost in revenue each day, Claire?
$200,000.
$200,000 per day. I'm in the wrong business. Aren't we all in the wrong business? If you can lose $200,000 per day, it's probably a pretty good business when you gain, right? So infer what key arguments on each side of the case are. So why does Creekstone think it should be able to conduct its own testing, and why did the USDA say nah to that? Barrett?
Because most of the time, U.S. Government departments don't feel reliable for mistakes.
There's that. That's
So
part of it.
If something happens in the company, will most likely push the blame to the USDA.
There's
And
that.
say that they miscalculated it, and they don't want to put a blame for the
Okay.
mistakes.
Anybody have anything else to add to it? So basically, Creekstone wanted to test every animal for BSE and to meet the demands of foreign buyers like Japan and assure consumers of safety regarding market access lost after this first case came in. But the USDA opposed it because it argued that their tests weren't scientifically warranted for 100% screening, especially in younger cattle, and that private testing would create a misleading false sense of safety, undermining confidence in the overall U.S. system, and also inciting the Virus Serum Toxin Act, which was a specific governmental situation here, to control test kit sales. The USDA claimed and approved rapid tests were only licensed for surveillance, right? Just who has it, right? and couldn't scientifically guarantee 100% safety in all animals, especially those under 30 months. Do you have a question?
Yeah, not really in class, but would this like disease actually make the meat of the cows like unsafe to consume? Or
No, who can answer that for her? How specifically, what was the problem? Yes.
The brain.
Brain. Okay. So how is that an issue for humans? Barrett.
Brain's the only thing you literally just.
No, I mean, that's true. Let me rephrase that question. How would an infected cow brain cause a problem when humans are consuming the cow? Yes.
You get what the cow.
You'd make it with the cow head. Anybody else? So it also comes to the point of when we do stuff like ground beef, we tend to take the beef that we're using to grind up, and it's not the inside of a muscle. It's come in contact with whatever else around it, which may include nervous tissue. So it's nervous tissue in general, not just the brain. Nervous tissue. Nobody's like, doing the whole eating cow brain. I mean, I'm sure somebody somewhere does, but we generally don't in our culture, like, make cow brain stew and eat it. But there's nervous tissue throughout the rest of the cow that can be impacted, and then you eat it. And does anybody remember what is the disease that people got from mad cow? What was it called? It was a variant Creutzfeldt-Jakob disease. Do you remember that? Creutzfeldt-Jakob is another prion, and they called it variant Creutzfeldt-Jakob because it was similar, but it came from a different source, right? And then what was the one that hit that isolated tribe? What was it? Kuru, right? And how were those people getting kuru? Do you remember this one?
They were eating the brains of the dead to
Yes.
honor them.
Yes, it was their culture. When someone died in their culture, it was better to eat their body than to let bugs and animals eat their body.
Are they gone?
No, the Fori are still there, I believe. But they stopped eating their dead because they had such a high incidence of kuru because people were dying, and it was predominantly women and children. But then there were men that would get it, and it would be because they'd had that long incubation period. But it was because the women and the children ate the brains, and then the men ate other body parts. That was just their culture. But they since learned maybe not such a good idea, and they don't do that anymore. Yep. All right. So now we're going into protist classification. Let's talk about, oh, by the way, I'm wearing diatoms. Yeah. You know me, I'm always wearing something fun. I'm wearing diatoms. So protists are any unicellular eukaryotic organism that is not an animal, plant, or a fungus. So if it's not a plant, it's not an animal, it's not a fungus, and it is a eukaryote, it is a protist, right? Now, while it's likely that protists share a common ancestor, the exclusion of other eukaryotes means that protists do not form a natural clade or group. Thank you for coming. Thank you. Thank you. Goodbye. 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 Hope to see you again. All right, let's see. Protists are a very diverse group of organisms. So the first prokaryotes, many species of protists are more closely related to plants, animals, or fungi than they are to other protists. So let's see. We have some that are more similar to plants. And this would be green algae is a great example of this. They are the closest relatives to land plants. Evidence shows they evolved into land plants at least 475 million years ago. Some biologists include green algae in the plant kingdom. Green algae are split into two groups, chlorophytes and charophytes. Both groups contain single-celled and multicellular algae. So charophytes are most closely related to land plants, and chlorophytes are found in marine, freshwater, and land-based ecosystems, but are most commonly found in shallow freshwater. So if these are most closely related to plants, you're going to expect these to be autotrophic, at least to a degree, right? And then those that are more similar to animals, protozoa is an informal term for a group of single-celled eukaryotes, either free-living or parasitic. What's a parasitic relationship?
Where one benefits and the other one harms.
Right. One benefits, the other is harmed, right? And feed on organic matter, such as other microorganisms or organic tissue and debris. Historically, protozoans were regarded as one-celled animals, but we know better, right? because they often possess animal-like behaviors such as motility and predation. They lack a cell wall, and they are found in plants and many algae. Historically, protozoa were divided into 4 big groups: amoeba, flagellates, ciliates, and sporozoa. And in some ways, this has to do with their means of locomotion. And then lastly, the ones that are most like fungi. Slime molds here in particular, we'll be talking about. Fungus-like protists are molds. They are absorptive feeders on decaying organic matter. They resemble fungi, and they reproduce with spores, as fungi do. In other ways, they're quite different from fungi and like other protists. For example, they have cell walls made of cellulose, whereas fungi have cell walls made of chitin. What is cellulose? Anybody remember what else we call it in our common day-to-day?
Fiber.
Fiber. Yes, Barrett?
Oh, that's something. I read up on this thing where there were time people went back to Chernobyl nuclear power plant
Yeah.
recently, and they found a fungus that was feeding off the radiation.
Yeah, that's scary. But yeah, So cell walls are composed, in plants are composed of cellulose, right? And cellulose is what we also call fiber. But the cell walls of these types of protists that are the slime molds is composed of chitin. Chitin is what composes the cell walls of fungi. It also composes the exoskeletons of arthropods. Interesting material, chitin. So like other protists, they have complicated life cycles with both asexual and sexual reproduction. They are motile during some stages of their life cycle. 2 major types are slime molds and water molds. Anybody ever seen the pink stuff that grows in toilets sometimes? Probably all gotten it at some point or another. Unless you clean your toilet like every 3 days, you may see it. It's like a pink, that's a slime mold. So slime molds can be all kinds of fun colors, actually. Pink slime mold that people get in their toilets is probably the most common one you might see. So protist ancestors and descendants. All eukaryotes are descended from early protists, but modern protists are very different from their ancestors. Like other eukaryotes, they have been evolving over the last 2.5 billion years. So we all came from some protist form, all of these higher life forms, but the current ones are very different from their ancestors. So structure and function. So here's your paramecium, your euglena, and your amoeba, what you were looking for in lab, right? Most protists are motile, but different types of protists have evolved varied modes of movement. Protists such as a euglena have 1 or more what that they use to whip and move around. What's the euglena have that moves with? Yes.
Flagellum.
Flagellum. Yep. Or a flagella if it's more than 1. Flagellum if it's 1. Paramecia, your plural of paramecium, are covered with tiny rows of what that they beat to swim? Caitlin?
Cilia.
Cilia. Very good. Other protists, such as amoebae, it's technically amoebae, although I think amoebas is pretty much now vernacular, form cytoplasmic extensions called what? What do we call them? Yeah, go for it, Lucas. Pseudopods, okay? Anywhere on the cell, and they anchor the pseudopod to the surface and pull themselves forward. Some protists can move toward or away from a stimulus. And what do we call a type of movement toward or away from a stimulus? Do you remember? Caitlin, do you know?
Spores.
No, not that one. Good guess, but starts with a T. Has an X in it. What is the thing you take if you're in a big city and it's yellow and you need to go from one place to another and you go, hey, pick me up. What's it called? A taxi, right? And a taxi is a form of locomotion. Helps you move, right? Taxis is movement toward or away from a stimulus. Oh, look, there's the same word again. I love it when these words come back and you're like, I know that word. That's not taxis more than one. That's just not plural of, not taxi, it's taxis, right? Protists accomplish what movement towards light? What's it called if you move toward or away from light? You've got taxis. That's going to be half of it. What do you suppose the other half of that word will be, Isabella? You got it. Phototaxis. Very good. So phototaxis would be movement toward your light by coupling their locomotion strategy with light sensing organ, like our euglena had the eye spot, right? So protists reproduce by a variety of mechanisms. Most undergo some sort of asexual reproduction. What produces two daughter cells? What type of asexual reproduction produces two daughter cells? Eunice?
Mitosis?
Not mitosis. That would be for cell division. If we're trying to get reproduction. Yeah.
Binary fission?
Binary fission. That's what we want. So binary meaning two, fission meaning to divide. Divide in two, binary fission, we get two daughter cells. Some protists, such as slime molds, exhibit what? And simultaneously divide into many cells. If binary fission is dividing into two, then multiple fission is dividing a lot at one time, right? Making lots of yourself from one original organism. Others undergo, what's this process where you get little tiny buds that pop off and grow all the way to the size of the parent? Answer's right there in the phrase. What's it called when they grow little extensions that bud off and make little copies of themselves?
Division?
Not division.
Budding?
Budding. There it is right there. She's like, is it budding? Yes, it's budding. It's not as hard as you think. Budding, very good. Produce tiny buds that go on to divide and grow to the size of the parent protist. Fungus-like protists were once classified as a fungi because they produce sporangia. A sporangium is a structure that contains spores for asexual reproduction. So what involves meiosis and fertilization? What's this process? This is sexual reproduction, right? It's going to involve meiosis, fertilization, parent protist, offspring protist in this case. And some protists will have the ability to switch, a lot of them do, to switch between asexual reproduction and sexual reproduction. Why would you want to do that? Why would you want to have the ability to reproduce both ways, Drew?
To increase genetic diversity.
Through which method? Sexual reproduction is going to increase genetic diversity. Very good. And why would you want to be able to reproduce asexually?
You can't find a mate.
You can't find anybody to hang with. You is alone. You is all alone. So yes, it's going to help you if you are a lonely protist to be able to reproduce asexually. Oh, I'll just bud, or I'll just binary fission. Sorry for me. And then you have others that are going to reproduce sexually. But their means of reproducing sexually is definitely different. And one in particular mainly occurs in ciliates here, and it's a process through which your nuclei and your gametes fuse together and form what they call a zygotic nucleus. Basically, paramecium, like another ciliates will do this, but they'll poof into each other, and they kind of dissolve the line between their membranes, and they sort of exchange genetic information. Anybody know what that's called? Conjugation. Conjugation. So sexual reproduction is often associated with periods when nutrients are depleted or environmental changes occur. Sexual reproduction may allow the protist to recombine genes and produce new variations of progeny that may be better suited to surviving in a new environment. Protist life cycles range from simple to extremely elaborate. Certain parasitic protists have complicated life cycles and must infect different host species at different developmental stages to complete their lifestyle. Some protists are Some protists are unicellular in the haploid form and multicellular in the diploid form, a strategy employed by animals. Other protists may have multicellular stages in both haploid and diploid forms, a strategy called alternation of generations, and is also used by plants. You'll see this in a lot of non-vascular plants. We'll have alternation of generations. So now let's talk about some ecology of protists. Autotrophic protists. Protists that perform photosynthesis are going to be autotrophic, right? The position of the photosynthetic protists at the base of the food chain makes much of diversity of aquatic life possible. They feed fish and whales. They support coral reefs. They provide shelter to marine life. In areas where sewage is dumped, these protists help recycle waste. They're super important. However, when the amount of waste is excessive, you get algal blooms into enormous masses, right? This is what we had. Remember when we had our chlorella vulgaris and we added high nitrogen fertilizer to it and we got suddenly a whole lot more, right? We got a whole lot more of it growing and you can get an algal bloom. What is the battle cry for University of Alabama?
Roll tide,
What are they talking about?
rolling tide.
What tide? Does anyone know? What are they? They're the what? What's their They're the crimson tide. What is that? Nobody knows?
I mean,
So-
not good.
It's a reddish algae that will bloom like this and turn the water red, therefore crimson tide. It's also very toxic. It will kill you. Therefore, they pick crimson tide. Ooh, we're dangerous. Personally, I'm way more afraid of yellow jackets. Even I'm not even a Georgia Tech fan, but I'm way more afraid of yellow jackets because you can avoid going in the water, but the yellow jackets will come for you. At least in my experience, they do. They're Satan's minions. All right. So we've talked before, and I've said to you that obviously deforestation and forest fires are bad for a number of reasons. So forest fires are part of a cycle, and you expect them to happen. They're actually part of nature, and as long as they're not like completely out of control and like some dude like California a year or two ago, that was really bad, that kind of situation. But you'll see forest fires that take place in the Amazon so often, and people will panic because they'll be like, the lungs of the earth are being destroyed. And I think I mentioned this to you guys before, but that's not the lungs of the earth. You definitely don't want it to be destroyed for a number of reasons, but you don't have to worry about running out of oxygen if a chunk of the rainforest burns. It's not good, but you're not going to run out of oxygen because phytoplankton is doing that for you. We got Hank here. We love Hank, right? Everybody loves
Thanks
Hank.
to the Monterey Bay Aquarium for partnering with
Love
us on this
Hank.
episode of SciShow. They hope you
He does have
ton of
cancer.
fun watching.
He has cancer. It's been rough.
Right now, there are incredible creatures in the ocean with the power to reshape the planet's atmosphere. After all, they've done it before, and you've probably never even seen them unless you've looked at seawater under a microscope. I'm talking, of course, about phytoplankton, the microscopic critter that almost all life in the oceans and arguably on land depends on. Phytoplankton is a catch-all category for all the tiny aquatic organisms that can turn sunlight into food via photosynthesis. And there are all kinds of different organisms included in it, from cyanobacteria to whip-tailed dinoflagellates to armored diatoms, which look amazing under a microscope, by the way. Diatoms are essentially algae that build little houses out of glass, or silica to be precise, and they come in a stunning diversity of forms. In fact, if you've ever spread diatomaceous earth around your garden or your house, you were sprinkling fossilized diatoms. There are also coccolithophores, which can look kind of like a soccer ball covered in frisbees thanks to their teeny tiny shells made of calcium carbonate. In fact, England's famous White Cliffs of Dover contain the fossilized remains of countless coccolithophores. And in today's ocean, the smelly compounds they produce help give the sea its unmistakable scent, a smell birds like albatrosses use to find their meals. The compounds that make those smells can also travel up high into the atmosphere to help clouds form and grow, making them more reflective and helping to keep us down here a lot cooler. And dinoflagellates are also very cool, don't want to leave them out. They have two tail-like flagella that help them swim around, and many can produce their own light, which makes for some really magical effects. But too many of them in one place can create a harmful algal bloom, a phenomenon you might have heard referred to as red tide, which is just one example of how these super teeny creatures can have a big impact on the rest of us, even us landlubbers. No matter what form phytoplankton take, they are unbelievably important to all life on Earth. First off, they're the foundation of the marine food web, underpinning everything from anchovies to whales. In fact, with a wee bit of help from seaweed and aquatic plants, they managed to support life in the ocean despite being outweighed by roughly 5-to-1 by organisms that don't make their own food. And if that's not enough, they are also atmosphere-altering machines. They consume about as much carbon dioxide as all of the world's forests and other plants combined, pulling roughly 11 gigatons of carbon from our atmosphere every year, which is especially impressive when you consider that by mass, there's about 450 times as much plant life on land. Plus, phytoplankton account for more than half of the planet's yearly oxygen production. But even more importantly, they're kind of the reason we have oxygen in the atmosphere at all. Back about 2.5 billion years ago, near the boundary between the Archean and the Proterozoic eons, the Earth was a very different place. There were green seas and orange skies, and the atmosphere was likely a mix of gases like ammonia and methane that would immediately kill us. Then cyanobacteria evolved and ran rampant, pumping out a huge amount of oxygen in what's now called the Great Oxidation Event. All that oxygen helped turn the ocean and skies blue and likely jump-started the evolution of complex life. And then around 540 million years ago, phytoplankton did it again, exploding in numbers and bringing us up to more or less the levels of oxygen in the atmosphere we have today. Paleontologists think the boom may have occurred thanks to nutrients weathered out of rocks by giant continent-sized glaciers. A more recent idea is that it happened because the plankton evolved a more efficient internal machinery related to photosynthesis. Either way, this set the stage for life to fully spread throughout the ocean and onto the land. Since then, they've calmed down on the reshaping the entire planet front and adopted more of a stabilizing role. For example, paleontologists think the evolution of calcifying plankton balanced ocean chemistry and helped protect the seas from mass extinctions. Before about 80 million years ago, the ocean was not a super stable place to be. Changes in the chemistry of the atmosphere from events like volcanic eruptions often caused deadly swings in seawater pH. Then some lineages of plankton, including those ball-like coccolithophores, figured out how to make shells of calcium carbonate. Soon, they took over the open ocean. And over a relatively short period of time, so many of their shells sank to the depths of the oceans that they created a vast, thick layer of calcium carbonate ooze on the seafloor. That ooze acts as a chemical buffer in the ocean, a natural braking system on runaway acidity. See, the shells are solid at a more neutral pH, but as the water gets more acidic, they start to dissolve, releasing carbonate ions. Those chemically react with the surrounding seawater to push an acidic pH back towards neutral. Now, there had been a minor version of this in the shallows, thanks to mollusks and other large-shelled organisms, but it took phytoplankton to make it ocean-wide. And the end result was a more stable ocean chemistry, which meant marine life became less vulnerable to things like volcanic eruptions. Even today, this chalk layer is helping buffer the acidifying effects of all the carbon dioxide that we humans are pumping into the atmosphere. Though there's only so much it can do. When we consider everything that lives in the ocean, from majestic whales to bountiful coral reefs, these little unassuming phytoplankton are easy to overlook. But if you look at the history of Earth, it doesn't take long to realize that though they are small, they are also mighty, and they deserve as much fascination, love, and protection as any other living thing on this planet. Thank you again to the Monterey Bay Aquarium.
Okay, now, holding some of those thoughts, I meant to put this in the slideshow, but I forgot. I'll show it to you. Come around and show it. This is a photo that I took at the Museum of Natural History when I was there a couple weeks ago in New York. And that's a sedimentary rock. Notice the red line. See the big red line? Okay, you see the big red line in this rock? I'll see it back here. Oh, see the big red line in this cross section of rock? What's the big red line? Anybody know what it is? No?
Solidified red stuff.
Solidified red stuff, yeah. That is the great oxidation. That is when you had phytoplankton evolve, because it suddenly started producing a lot of oxygen. And the oxygen came in contact with what element do you suppose in the rock? Iron. And then we get this big red band. That can be found all over the earth in various places, this nice big red band, where you know that suddenly now we're getting oxygen in the atmosphere. So that is the evolution of phytoplankton right there, like on the rock. And I was sitting there saying to Jason, oh, cool, when was this? Oh, I heard that. And this guy comes up to me, this guy, you know what that is? I was like, yeah, I think I do. So we talked for a minute. And then he asked me if I was a geologist. I'm like, no, I just teach all the sciences, like all the sciences. It was kind of funny.
Why the sky orange but the seas blue? I thought the seas were blue because they reflected the sky.
The sky has a different atmosphere now. So
But how did they not reflect orange?
How did they not reflect orange? Oh, well, no, it's not just, they're not reflecting. It's not, that the ocean is blue and the sky is reflecting it. It has to do, we talked about it in physical science, but it has to do with the wavelength of light that's making its way through our particular atmospheric composition. And that is what is being reflected is blue. When the atmospheric composition was different, it was orange that was reflected, not necessarily the water below. So yeah.
What would it take for the sky to be red?
That happens at sunset all the time, right? So that has to do with angle of light and atmosphere. Right. And there can be certain components in the atmosphere, like whenever there's a volcanic eruption somewhere, or even fires, big fires, wildfires, it can change the sunset for a vast number of viewers, depending on where they're located. So the entire sky, it would have to be a combination of those things. It would be more difficult because the angle has to be right. And we get the angle when the sun is lower on the horizon. But yeah, I mean, it could happen with the right combination of things happening all at once. But I think I'd be a little afraid if it happened in the middle of the day. I'd be like, oh, now I'm in Vecna's land or something because they had red sky in there. I don't want to be there. All right. So now let's talk about heterotrophic protists. Some heterotrophic protists engulf and digest their food, while others live by absorbing molecules from the environment. Now that's kind of what your amoeba might have looked like should you have been lucky enough to get one under your frame. That's not chloroplasts. Those are food vacuoles. That's what it's eating. So amoebas capture and digest their food surrounding a cell or particle and then taking it inside of themselves to form a food vacuole. A food vacuole is a small cavity in the cytoplasm that temporarily stores food until they manage to chemically digest it. Paramecia and other ciliates use their cilia to sweep food particles into their gullet, an indentation on one side of the organism. It's right here on this guy. And then slime molds and water molds are important recyclers of organic material. At one stage of their life, some slime molds fuse to form large cells with many nuclei. So basically they lose their cell membranes, and they just make this massive cytoplasm with little nuclei floating in it. And it's known as a plasmodium. So we're going to watch something very cool here in a second. So fungus-like protists, like slime molds and water molds, obtain their energy by decomposing organic materials, and as a result, are important for recycling nutrients. They can be brightly colored, live in cool, moist, dark habitats, like that pink mold in your toilet. Slime molds are classified by either plasmodial or cellular, and by their modes of reproduction. So the plasmodial ones, like I just said, they drop all their cell walls, and it's just like a massive cytoplasm with nuclei scattered amongst it. And then the cellular slime molds exist as individual cells during their feeding stage, and they move like amoebas, as well as engulfing their food along the way. Now, this is one of the coolest things. I love when this lesson comes up for this video. This video alone makes the lesson. All right, this is a slime mold. Slime molds can solve mazes. For real. No brain, no ability to think and make decisions. They can solve mazes. But they can also do something else, which towards the end of this video you'll see. And it's super impressive.
At Oxford University in England, Dr. Mark Fricker is one of a team of botanists and computer scientists studying for a slime mold called Fusarium polycephalum. For years, slime molds have fascinated scientists with their ability to navigate the quickest route through a maze to get at some food. But scientists started to wonder if the slime mold could do more than just perform clever tricks. Here, Mark is recreating an experiment he worked on with colleagues at Tokyo University. A blob of slime mold is surrounded with a pattern of oat flakes, an irresistible treat for the slime mold. What happens next is recorded by a time-lapse camera.
So the slime mold starts as this blob in the middle, and then it's going to spread out and colonize the whole of the dish, trying to find all of those oat flakes that we positioned earlier on. So connect each of those food resources but some of those links will disappear and some of them will be strengthened.
What the slime mold has done is truly remarkable.
The way we set out those oat flakes was not entirely random. The central blob is supposed to represent Tokyo, and if we positioned all of the oat flakes on the major cities around Tokyo, so what we're now looking at is not just the slime mold exploring a random distribution of oat flakes, it's actually trying to map out the region around Tokyo to see how the slime mold would connect all of those cities to compare against the way humans have connected it with their own network.
When you compare the local Tokyo rail system and the network that the slime mold has come up with, the two designs are remarkably similar. In just a few hours, the slime mold has done what skilled engineers took years to achieve. It has found the most efficient way to link together multiple locations, every bit as effective as the well-organized Tokyo rail system.
The slime mold doesn't have a brain or any way of calculating this, but has managed to produce a network with very similar properties.
All right, is anybody else as fascinated by that as I am? Because that floors me, that a slime mold can basically create a near identical copy of the Tokyo rail system because it finds the most efficient route between its little sources of food. And all the other little routes disappear and it strengthens the more efficient ones.
The more efficient route might be different for humans though, because like
Topography. Yeah, topography and stuff. And you could see that there were some places where it was slightly different. But if you were doing as the crow flies, that's exactly what it did, right, Jordan?
Just those guys is like
Let them do central planning from now on. Like they might do better than some of our current people are doing, because that was pretty impressive, right? Like it looked almost exactly the same in the end. And I like how it creates all these little branches and then almost forgets about the ones that aren't doing very well. It just strengthens the very efficient ones. Really interesting. All right, so nope. Symbiotic protists. Some protists have symbiotic relationships with other organisms. What symbiosis mean? Isabella? Both organisms are going to benefit from the relationship. Very good. So, Trichonympha has a mutualistic relationship with termites. It lives in their digestive system and helps them digest wood. Did you know that termites cannot digest wood, even though that's all they eat? A termite by itself? No, it's just snacking on something it can't digest. That's a little weird when you think about it, right? Now, we are going to use some termites in lab in several weeks to see their behavior. It's really kind of interesting. We're going to use, see how they behave with certain chemoreceptors and if it changes their pattern of movement. It's really fun. It's a fun lab. Other protists are parasitic and can cause disease. Trypanosoma causes African sleeping sickness. Nobody wants that. The Protist plasmodium causes malaria. Nobody wants that. Not good. Now let's talk about fungi. Alrighty. What are fungi? Fungi are eukaryotic heterotrophs that have cell walls. The cell walls contain chitin, which is a complex carbohydrate. What other organism did I tell you contained chitin? Remember? Yep, specifically arthropod skeletons are also made of chitin. Very random, right? We've got roly-poly, not roly-polies because they're not frogs, they're crustaceans. Okay, grasshopper shells or ants and mushroom cell walls. Same material. So most fungi are composed of thin filaments called hyphae. The fruiting body of a fungus, such as the above ground part of a mushroom, is the reproductive structure that you see. It grows from many hyphae tangled underground in a thick mass called the mycelium. The mycelium is the main part of the fungus. You don't see it because it's underground. If you've ever dug around like a rotting log or whatever, you might see the little hairy, like little white, hairy, fuzzy looking things that kind of grow everywhere. That's the mycelium. That is the organism. The mushroom is only the reproductive structure. So when you see mushrooms outside, you're only seeing the reproductive structure of that particular fungus. The rest of it, the main part of it, is still under the ground. Most fungi reproduce both asexually and sexually. Asexual reproduction can occur when cells or hyphae break off and begin to grow on their own. Some fungi also reproduce asexually by means of spores, and we're going to do an activity with spores before you leave today. Should be fun. Hopefully it works. Most fungi can also reproduce sexually. Spores are produced in structures called sporangia. Many fungi have a negative or a minus and a plus type. We don't say male and female really in this case, but we say a minus and a plus type. And they can reproduce sexually by fusing their nuclei when they meet. All right, now, trees in the forest are communicating with each other through fungus mycelium. That's how they talk. And they share all kinds of information. They share information about, hey, I have a parasite. They share information about water levels, nutrient levels, and that's all because of mycelium. So when you look out there, you may see, oh, here's a tree and here's some fungus, and you may think of it as being interconnected, not interconnected, but it is very, very interconnected. And this video will show you just how interconnected that mycelium is to all the trees, and in particular trees, hub trees.
Is car insurance.
Yeah, yeah.
At your own pace. Tired of big monthly payments?
Never heard of it. Okay.
The idea of talking trees has been capturing the human imagination for generations.
My God is worse than my life.
Okay, so maybe they don't talk to us, but it turns out that trees can talk to each other.
Trees are speaking to each other.
But that does beg the question, what do trees have to talk about? And can you learn to speak their language? Underneath the soil, a vast and interconnected network of life links the trees through their root systems. But they can't talk to each other without help. The whole process starts with hub trees, the oldest and tallest trees in the forest. Hub trees have greater access to sunlight, and through the process of photosynthesis, end up producing more sugar than they actually need. Underground, fungi need sugar to survive. Most of their bodies are made up of a mass of threads called mycelium. They grow within the root system of trees to absorb the excess sugar. In return, the mycelium provides the tree with the nutrients it needs from the soil. This symbiotic relationship is known as mycorrhiza, which stems from the Greek words for fungus and root. These tree-fungi relationships connect the trees in the forest together, forming an underground communication network to exchange water and nutrients, nurture their seedlings, and even send warning signals when under threat. So, how many trees are really talking to each other? To get a better picture of these forest relationships, a team of researchers used DNA analysis to map a fungal network in a patch of Canadian forest. Remarkably, they found that one tree was connected to 47 other trees. Their models also showed that when hub trees were removed, it would cause more connections to be lost than if trees were simply removed randomly. Studying these kinds of underground exchanges will play a vital role in creating stronger, more resilient forests for the future. So even though we might not be able to talk to trees, at least we can still keep trying to understand their language. Who knows what they might say?
So this is like nature's forest telecom network, really. I mean, this is how they are communicating with each other. And those hub trees are essential because you saw when they removed the hub tree, you lost a lot of connections, right? So obviously another tree will eventually take its place, but that's going to take time for that tree to get tall enough, big enough, and be the one that establishes the connections. Yeah.
Since they can send like warning signals to each other, but what exactly are the trees gonna do about that?
So that's interesting. So trees can organisms, plants can respond, right? They can produce certain chemicals that protect them, if they're under attack by a parasite or whatever. And I think that may be the goal. I'm not entirely sure of all the details, but I imagine it can help prepare another tree or another plant for what's coming, whether it's a lack of nutrients or a parasite or too much water, too little water, and allow them time to sort of restructure the way they're functioning in order to best handle that situation. But that's pretty wild that all happens through fungus, mycelium, and not even anything that you can see. It's just all underground, big network. And it reminds me of a telecom map. I don't know, the telecom guys in the room, does it remind you of a telecom map? You saw it?
Sure.
Yeah, sure. Okay, you know what that is? The fairy ring. Anybody ever seen one? You're lucky if you see them. I love it. Whenever I see them, I get very excited. Sometimes it's not a complete circle, it's just sort of an arc. But a fairy ring happens because the mycelium, the center hub of the mycelium is in the middle. And it spreads out and it pops up little fruiting bodies, little reproductive structures. And it just so happens to do it in this nice circular pattern because the mycelium is centered. So it extends out and makes all these little growths around the outside, and you get these mushrooms around the outside that appear, and it can change the grass. There's a lot of nutrient exchange going on, so it's either good or bad for the grass. So sometimes you'll have really lush grass, a very green grass in the area of where the fruiting bodies are located and in the center, and other times you'll have dead grass, depending on the situation. It can depend on the given situation. Why do you think it's called a fairy ring?
Because the little fairies come out of the ground and have their meeting.
That's exactly why it's called a fairy ring, because people envision cute little fairies sitting on the little toadstools and having their little meeting while they're all sitting around in a circle. Isn't that cute that they sit in a circle? How communal of them. Like, how nice. It's a cool image. Fairy rings are neat. I've got a neighbor who I drive past who gets a lot of them in the spring and fall when it's not too hot out. It's pretty cool. Yeah.
You gotta put a round table in there.
Yeah, you should. Put a little table in the center. You need a little fairy with a little gavel. Call the meeting to order, right? Very cool. And remember that the mushrooms and toadstools and whatever you're seeing, that's just the reproductive structure.
This family that I babysat once, they have like a pretty big backyard with like lots of trees and stuff around, and they had a fairy ring in there.
Yeah.
And I taught like their 5-year-old how to make fairy houses and
Oh, fun. Yeah, totally. That's fun. Has any ever been to the fairy village off the Greenway, by the way? Recommend if you haven't been. I don't know what state it's in right now, but it's over by Halcyon. But there's some people have just stepped off the trail into this area by a creek and just built a tiny fairy village. People are always building and changing things there. It's kind of fun. It's cute. It's neat. It's neat. All right, so let's talk about ecology of fungi. Fungi do not ingest their food as animals do. Instead, fungi digest food outside their bodies and then absorb it. Many fungi feed by absorbing nutrients from decaying matter, and some fungi are parasites. Fungi help maintain equilibrium in nearly every ecosystem by recycling nutrients and breaking down bodies and wastes of other organisms. Now, if you leave fruit out on the counter too long, it's going to start getting fungus on the outside of it, right? Because that is the job of the fungus. Where does that fungus come from? Yeah, some of it's airborne spores, absolutely. Yep. So some of it is probably already on your food when you get it. It just takes a while to start really growing, right? Parasitic fungi cause serious plant and animal diseases. Fungal diseases in humans include athlete's foot, thrush, yeast infections, and the female reproductive tract. So you can get yeast overgrowth anywhere on you, and other kinds of fungus as well. Athlete's foot, right? Thrush is when babies get a yeast infection in their mouth, right? That can happen with babies. And some fungi form mutualistic relationships, which benefits both partners. Lichens are the great example of this. Everybody knows what a lichen looks like, right? Growing on the trees outside, right? So a lichen is a symbiotic association between a fungus and a photosynthetic organism, usually a type of algae. And the photosynthetic organism provides a source of energy, and the fungus provides water and minerals, and they live happily ever after as a lichen until they're eaten by a reindeer, right? And that's what they do. I love them. There's some of the lichens that look like moss almost, and others are flat to trees. They're really, really fun. Mutualistic associations of plant roots and fungi are called mycorrhizae, which we just saw in that video. And the plant's roots are woven into partnership with a web of fungal hyphae, and they use it to communicate even. All right, some mushrooms are bioluminescent and they're cool. And you can buy these online. There are some you can buy. There's one that the bioluminescence is orange. I forget what it's called, but it's supposed to be able to grow in this zone. And I've been very tempted, but I'm sure I won't grow it well. Yeah.
Can you like grow multiple of them from one? Like
Yeah, I mean they'll reproduce as they do. So these are particular green ones do not grow here, the one he's showing here, but there are some you can get that apparently will glow at night. Like I said, I've been very tempted to get them.
Hugo is car insurance.
Again with this
that lets
knockoff
you
car
pay at
insurance.
your own pace. Tired of big monthly payment?
Yeah.
Could never be done, though.
Right now, we're not sure why the fungi bioluminesce. But that's what we're trying to do right now. Let's figure out why. So organisms bioluminesce for various reasons, but it's probably all started out from just basic detoxifying processes that happen in the cells. Some of the antioxidants, as they break down, give off light. As these traits evolve, other organisms go on to gain additional functions, like fireflies use their light system for mating and communication between species. A lot of that also happens inside the marine environment. But right now, we're not exactly clear as to why mushrooms do it. The experiment that I'm performing out in the field is an ecology experiment where we are trying to understand the interaction of the mushrooms with insects. And so to do this, we're using fake acrylic mushrooms that we've produced in the lab and putting them out in the field covered in glue. Some of them have an LED light that has about the same wavelength as the mushrooms. So it's producing the same colored light. And so we're comparing the amount and the type of insects that are on the dark traps to the light traps. And by looking at the two sets of data, we can tease apart, which insects are attracted to the light and which insects are repelled by the light and which insects just don't care about the light. Through this, we can better understand the interaction of these mushrooms with their environment. It's very likely that the bioluminescence from the mushrooms has some ecological significance in the forest. Based off of our early results or early data, it definitely looks like insects are attracted to the mushrooms. Now, what role those insects are doing requires further study. Fungi are at this special moment right now where the research is really beginning to progress, and we're likely to figure out the mechanism within the next few years.
What do you think it might be? Just out of curiosity, what might it be? What do you think? Pest control? Mm-hmm. Could be thing. What do you think, Isabel? Some sort of symbiotic relationship with certain insects, yeah. So some insects are going to be attracted to that light. It's interesting. Maybe the insects are carrying something on their bodies that the fungus wants, or maybe the fungus wants the insects to take something from them. I don't know, but it's very interesting, right? Pretty cool. I'd like to see them actually glow in the dark. Mushrooms would be pretty neat. All right, so somebody define what's a protist. Caitlin. Single-celled eukaryotic organism that is not an animal fungus or a plant, and it's not a prokaryote, it's a eukaryote. Very good. Why are brown algae considered protists even though they are multicellular? Anybody have this one? Nobody has this one? They're more closely related to some unicellular protists than any other kingdom. All right, why do scientists no longer use categories of animal-like, plant-like, fungus-like to classify the protists? What you got?
Because they're inaccurate and they don't actually represent what they are.
They don't accurately represent. Yeah, they don't reflect the evolutionary history in particular of the protists. So most single-celled eukaryotes are classified as what? What do we have? Claire. There we go. Protists. Genetic analyses of protists indicate they belong to 6 different. Jordan. There we go. Clades. Very good. Protists were the first what? What were they? The first what? Eunice? Eukaryotes. Very good. Unlike most protists, which are unicellular, kelp has what kind of tissues? Blink, blink, blink. Kelp has differentiated tissues. So kelp, seaweed, right? You've seen it before. It's huge. It's under the water and it's big. But it's multicellular, but it has differentiated tissues. So it's interesting. Because it's different from a lot of like the guys we saw today, right? All right, so your diagram would look something like so. All right, plants are most closely related to which of the following groups? What do you have, Eunice? Red algae? Anybody have anything different? Or are we going with the red algae? Very good. Rhodophyta is red algae. Very good. Brown algae is a member of the clade I'm going to get this one. Chromoviolates, I believe, sorry. Which of the following is its closest relative? Caitlin?
D.
D. Very good. Which clade is the most primitive clade? Kenna? Hey, she says, yes, excavates. Very good. Where are you? There you are. Which statement is true? Caitlin?
A.
A. Plants, fungi, and animals all emerged from a common protist ancestor. So these are our ancestors. But obviously we have evolved drastically since then. I hope. Sometimes I wonder. Sometimes I wonder. And what are these guys right here in this image? Those are diatoms, right? They have the little silica, they make little silica shells around themselves like this, right? And they can be absolutely beautiful. They can be so pretty. Protists, ancestors and descendants. So true or false? First eukaryotes were archaea. True or false? Kenna?
True.
That one's not. You had it earlier. You said excavates, right? All right, so let's see. It's possible to find the earliest fungi by looking at modern protists. True or false? What do we think? False, he says? False. Modern protists have evolved very little from their ancestral forms. Everybody should know this one because we said it earlier. Asher.
False.
Yeah, they've evolved a lot. They don't look anything like they used to look. Today, there are as many as 300,000 known species of protists. Caitlin?
True.
She says true, and she's right. 300,000 species. That's a lot. That's known, exactly. The oldest known eukaryotic fossils are about 1.5 billion years old. Eunice?
True.
It says true. That's right. The first protists most likely evolved 3.6 billion years ago. What you got? False, he says. False, he says. Whoops, hang on. Plants, animals, and fungi most likely evolved from multicellular protists. What do we think about that, Jordan? Yeah, there we go. Like the kelp, right? Protists are more diverse than any other eukaryotic kingdom. What do we think of this one, Colin? Yeah, true. So more diverse than Animalia, Plantae, fungi. You got Protista, and it's going to be more diverse. All right, so looking here, certain ancient species of archaea evolved into ancestral protists, which evolved into plants, animals, fungi, and the many different groups of modern protists. Euglena are photosynthetic protists that live in fresh water. How would their photosynthetic properties help stabilize a pond ecosystem? Suppose they had all the other photosynthetic organisms, so suppose all the other photosynthetic organisms disappeared from the pond. How might their disappearance affect organisms that lived in the pond? Asher? A lack of oxygen. Drew? That's what you're gonna say? Okay. Oh, you have something else to add, Lucas?
There'd be no more autotrophs.
There'd be no more autotrophs, or yeah, there wouldn't, not those autotrophs in particular, at least. Yeah.
Everything would die.
Everything would die. Okay, so photosynthetic properties of euglena would help stabilize the ecosystem because they would utilize the carbon dioxide secreted by some organisms and provide oxygen for use by others. If euglena and all the other photosynthetic organisms disappeared from the pond, and all other organisms would eventually die, Asher, since the bases of all the food chains would be gone. All right, so now, what are pseudopods and how do protists use them, Eunice?
Pseudopods are used by extending temporary projection of cytoplasm.
Right, and what does pseudopod mean?
There are
False foot, yep. A temporary projection of cytoplasm used for feeding and movement. How do amoebas, there's the other spelling, capture and ingest food? Yes. The hug of death, as I like to call it. Yes, they are going to use their pseudopods to surround a food particle or cell and then take it inside themselves, ingesting it. The hug of death. What are cilia and how are they used by protists? Claire.
Like little tiny feet that are constantly moving, kind of like robots.
Yes, there you go. Short hair-like projections used for movement. So these are, you can look at what you drew. I'm not going to come look at what you drew necessarily, right? All right, so looky there. Remember what I said about rowboats earlier? There it is. A visual analogy compares structures used by cells for movement to boat oars. Explain why a series of oars is compared to cilia and why only one oar is used to represent a flagella. Well, it's pretty obvious. You've got all the oars together like cilia. They're all beating together in the same direction, and then they can change direction quickly, right? But they don't all beat randomly. They all go one way, change direction. And then when you've got your flagellum here, it's analogous to like a back and forth movement. It's almost like a rudder steers. They can kind of steer it a little bit, but mostly they just kind of move pell-mell, like whatever's going. It just flips all around and they just kind of barrel forward, right? All right, so how do amoebas reproduce? Which of those methods, Drew? Asexually, yep. By what method? Do we know? So these are, oh, sorry, that's the answer. These are going to be like a mitosis, like a binary fission type of situation. They're just going to divide themselves, right? And what was conjugation? We talked about conjugation. Eunice?
Sexual process in which two organisms exchange material.
Yes, a process that allows ciliates to exchange genetic information with other individuals, but not like a true sexual reproduction where you have haploid makes diploid, right? It's kind of a different exchange, but it's very, very similar in the end result, right? So with the large population, how does conjugation benefit protists, Caitlin? Genetic diversity is what you want. Yep, helps create and maintain genetic diversity. What occurs in the process known as alternation of generations? Do you know this one? Do you? Not sure?
Can't try.
Jordan wants to try?
The swapping between asexually and sexually.
Yeah, so alternation of generations means sexual reproduction in which the reproductive cycle is a means of sexual reproduction, in which it switches back and forth between haploid and diploid stages. So not so much sexual and asexual, but haploid and diploid stages. And you will see this when, like I said, when we get to plants, we get into our like very primitive non-vascular plants. You'll see this exact sort of alternation of generations that occurs in nature with them and the way they reproduce. So we have a little flow chart. It says conjugation begins when two paramecia attach to one another. Oh. when one paramecia meets another paramecia. All right, so what happens next? What has to happen in order for them to exchange information? So the next thing is there's meiosis of their diploid micronuclei to produce haploid micronuclei. So their nuclei in each of the individual paramecia is going to divide and make little haploid forms, all right? In each cell, 3 of the micronuclei disintegrate. That's fun. And then what happens is your remaining micronucleus in each cell divides by mitosis. Then you have the two cells exchange one haploid micronucleus from each pair. Whoop! And in each cell, the micronuclei fuse to form a single diploid micronucleus and a macronuclei that they disintegrate. So that's basically how they exchange genetic information. And then lastly, each cell forms a new macronucleus from its micronucleus. Cool. Plasmodium is the protist that causes malaria in humans. It is carried by mosquitoes, which transmit the parasite when they bite humans. In the human body, plasmodia causes red blood cells to break and release spores in the evening. What? Okay. Spores that can then, sorry, I thought I read that wrong. Spores can then travel through the bloodstream. How would this mechanism help spread the parasite through an ecosystem, and what methods might help prevent its spread? So in this case, mosquitoes bite in the evening. By releasing spores at that time, the parasite increases its chances of being ingested by the mosquito and carried to another host. Preventative methods may include staying inside in the evening, wearing bug sprayer clothes, with total coverage, sleeping under nets, and keeping windows and doors screened. There is a whole movement in getting mosquito nets to people that live in regions with high amounts of malaria. Mosquito nets are an inexpensive way to do it. You throw the net up over your bed at night and you don't get bitten while you sleep, and that's going to reduce your chance of getting malaria and dying from it. Now, does anybody know what chemical compound was used, or what chemical was used as sort of our main first line of defense against malaria? Do you know? That's a repellent. So that's a good one. That will repel mosquitoes. They don't like it and they'll go away from it. But do you know? Not arsenic. That would be fun. That would be dangerous to everybody else. So we used to use a lot of something called DEET, DEET, and that was used in a lot of insecticides. But too much of that can cause developmental problems. So we don't use it as much anymore. We still use it, but not on a grand scale. We were using it on a grand scale. And it was great because it was killing off all the mosquitoes, and that was fabulous. But then it created other problems.
What's that thing where pregnant women got bit by mosquitoes?
Zika. Yeah. And that happened right around the time of the Rio de Janeiro Olympics. And nobody wanted to go to South America because Zika was rampant. Did anybody know Zika virus or heard of Zika virus? The Zika was another thing that was being transmitted by mosquitoes. And when a pregnant woman was infected, it affected the fetus to the point that the baby was born with like a microcranium. Like these babies had these little tiny heads, this misshapen heads. They were developmentally delayed and handicapped and had all these problems. And I mean, we're talking like a baby with a head, like tiny little head. It was not a good situation. And that was not good news for South America when they were about to hold the Olympics in Rio de Janeiro. And the whole world was like, I don't know if I want to go down there. You have dangerous bugs. It's gross. But yeah, that was Zika was a problem too. Are you looking it up? Look up Zika.
Trying to look. I remember seeing.
Yeah, it was.
About that, but
yeah, it was pretty wild. It caused some definite issues. Like for example, here. Yeah, here you go. So like, this was the result of Zika virus biting pregnant women. Like poor little baby. Like that's not good. That's really not good. So yeah, all kinds of issues. You don't want to get bitten by mosquitoes in general. They can give you all kinds of things.
Pants,
they still get you
over my pants.
Yep.
And literally they couldn't get me anywhere, and I still came home with several bug bites.
They are more attracted to certain blood types than others. I think it's O that they like best. Mm-hmm. And they also are attracted to carbon dioxide that you exhale when you talk or just breathe.
Don't.
Mm-hmm. They make some mosquito traps that emit carbon dioxide to lure them, and then they get sucked in into like a little fan. I'm one of those people that it's like if there's a mosquito in a 5-mile radius, it's gonna find me. Like it is going to find me. And And I react huge. Like I get big, giant. Yep, so fun. All right, autotrophic protists. How do autotrophic protists make the diversity of aquatic life possible? Think back to Hank. So these are the base of the food chain. What are phytoplankton? They are little guys. Yeah, sure. Yeah, all that stuff. Yep. These are the small photosynthetic organisms found near the surface of the ocean. Many are autotrophic protists. Yep. These are the guys that put all of the oxygen, most of the oxygen into the atmosphere. And they do help chemically balance the ocean. How do protists maintain homeostasis in coral reef ecosystems? Anybody know? So in this case, they the particular red algae type support coral reefs by providing much needed nutrients for coral animals. Red algae also produces mineral corals need to form the reefs. You know, coral is an animal, right? Even though it like looks nothing like an animal, it's an animal. Kind of one of those hard to believe ones, like sponges. Like, what am I looking at? Uh-huh. So how can algal blooms be harmful? Yes, Eunice?
There's too much algae.
There's too much algae, which causes what kind of problems? Do we know? So it can deplete water of nutrients. Decomposition of the dead algae can rob water of oxygen. It can choke resident fish and invertebrate life. And some of them release toxic chemicals, which can make you very sick too. What is the function of a food vacuole? What's the purpose of a food vacuole? To store food. And what else? What else does it do? Yeah, to digest the food, to store it until it can be digested, and then yep, they just kind of break open and it's digested. So a paramecium would look like so. This is hopefully what you were trying to see today. Maybe not. If you were lucky, you got some. And it may have been hard to get, yeah. Oh, did it do its thing? It may have been hard to get one and then also get one still enough to be able to diagram it. We do have some on prepared slides that are a little easier to see. All right, so then what are slime molds? Yes.
Recyclers of organic material.
Yeah. So slime molds are heterotrophic protists. They thrive on decaying organic matter. By what process are haploid spores made by a water mold? And where does that process occur? Anybody know? So spores are made by meiosis inside the sporangium. What structure does a plasmodium eventually develop into, and what is the function of the structure? Do you remember that the plasmodium is when all of the cell membranes disappear and you just have like a blob of cytoplasm with a bunch of nuclei? Drew, did you know?
Is that the sporangium with phosphorus production?
Yeah, that's what you got. So a plasmodium eventually changes into sporangia, which produce haploid spores. So true. And if it's false, change the word to make it true. In amoebas, indigestible materials remain inside a contractile vacuole. True?
False.
False. What should it be?
Should be.
A food vacuole. Yep, there you are. All right. Now, the gullet is a structure used by paramecium for reproduction. Reproduction? Lucas. Eating. Yeah, feeding. In a slime mold's life cycle, germinating spores release amoeba-like cells. That's true. Water mold grows on dead or decaying plants or animals. Is that true or false?
False.
False. What should it be? Water. Well, you have to change water molds. So that gets changed to, actually, that one's true. Sorry. So that one is true. They do grow on dead or decaying plants or animals, but they're very watery and gross. Yes. All right. Symbiotic protists, mutualists, and parasites. How does the protist Trichonympha make it possible for termites to eat wood? You know, Lucas?
Digest it.
Yeah, they do. Specifically, termites don't have the enzymes to break down the cellulose in wood. The protists in a termite's gut manufacture the enzyme cellulose, cellulase, sorry, which breaks down the chemical bonds in cellulose, which helps with that help of the protist partners, then termites can digest wood. So termites cannot digest wood without their little protist buddies living in their guts. What causes malaria? Drew? Yeah, and what's the organism, do you know? Plasmodium is the organism that does it. And yes, it's transmitted by mosquitoes. So here is a little flow chart of Plasmodium life. Dum-da-dum-da-dum, coming around, causing problems. Mosquito bites infected person, picks up Plasmodium gametes, Plasmodium sporocytes pop in here. Merocyte cells infect red blood cells and reproduce. Infected cells burst. Merocytes and gametes are released. Slime molds are heterotrophic protists that thrive on decaying matter. How would they help maintain homeostasis within their ecosystems? How do they benefit an ecosystem? Why is their role so important? Anybody? Drew?
If they recycle decaying organisms and they prevent waste buildup.
Yeah, so they help decompose decaying matter and plant and animal matter. They help recycle nutrients through an ecosystem, which benefits any ecosystem. Without them, there would be fewer nutrients being recycled and the ecosystem would be smothered by decaying matter. Why do scientists think that fungi are more closely related to animals than to plants? What do we think? Cell walls of fungi contain chitin. Similarly, insect exoskeletons contain chitin. So it's different from plants, right? Two types of hyphae are going to be one type has cross walls that divide into compartments like cells, and the other does not. Parts of a fungus would be labeled accordingly. If you had your little mushroom, your fruiting body's up here. This is your reproductive organ. Then you have your hyphae and your mycelium down underneath. What's the function of the fruiting body then? I just said it. It's what kind of structure? Asher? It's the reproductive structure. Yep. What's a fairy ring? We talked about it. Claire?
A circle of mushrooms.
Yeah. Fairy ring is composed of fruiting bodies of mushrooms that developed at the outer edges of a single mycelium. It forms because as time goes by, soil nutrients near the center of the mycelium become depleted. The fruiting bodies sprout only at the edges. So here is a diagram of this particular life cycle. You can check and see how you did there. Knew that was going to hit and make a noise. All right. How do fungi break down leaves, fruit, organic material into simple molecules? They're going to release digestive enzymes to speed the breakdown of those materials. How can fungi disrupt the homeostasis of plants? They can cause diseases such as, I love this one, corn smut and wheat rust. We got a corn smut problem. Just sounds wrong. Lichens and other mycorrhizae are both examples of what kind of symbiotic relationship. We talked about that. That is mutualism. How do plants benefit from mycorrhizae? How do fungi benefit? We watched a whole video on that. Fungi collect water nutrients, bring them to plant roots, as well as freeing nutrients in the soil. Plants provide photosynthesis products to fungi. And here is this diagram for you. You've got your algae and fungus together that form your lichen. Photosynthetic organisms provide the fungus with a source of energy, and the fungus provides the algae and the water, minerals, and protection. Fungus-like, oh, do I have that answer? Oh, yes, I do. A fungus-like chemical soaks into the ground and is absorbed through the roots of a plant with a fungal disease. How might this help the infected plants regain homeostasis? How might it damage the homeostasis of other plants in the area? The chemical would kill the fungus that was harming the plants, restoring them to homeostasis. It could, however, harm other plants in the area if it killed mycorrhizal fungi on their roots. All right, now we're going to have a quick little activity before we leave. So we're going to do this over in the breakfast room, but don't move yet. Let me make sure you understand what you're doing. So what we're doing is I have got some mushroom caps for you that I very carefully removed. Hang on. The stems and the little outer edges so that their gills are exposed. What lives in the gills of the mushrooms? Spores. Okay. This may or may not work. Sometimes it works. Sometimes it doesn't. It usually works better with darker mushrooms like portobellas or creminis. But it usually works with these guys too. So what you're going to do is there's a narrow table against the back window. You're going to take a blank piece of paper and you're going to write your name on it. And then you're going to flip it over. And on the other side, you're going to take, well, how you can divide up those mushrooms between you, probably between 1 and 3 each. All right. And you're going to put them gills down on the paper, and you're just going to leave them there. Now we have to fit everybody's paper on there, so you may have to gently slide those papers around, but we don't want to lift them up and move them because the junior stuff has already been sitting there for a while. So next week when you come to class, I'll have whatever spore print is available. It should make a little print. It probably won't be this dark in color because those mushrooms were lighter. The darker ones didn't look good when I was at the store, and I was like, these are going to be yucky and I don't want to use them. So I'm going to leave them for a day or two. And then I will toss the mushrooms and give you your prints back next week. And then you get to take it home and turn it into something and then bring it back and we'll show it off. Okay? So let me clear that, move this out of the way a little bit. Let's move down here to the middle table. The mushrooms over here. Have some more. There's a bunch of skin floating around. There you go. I'm going to scoot these a little more out of the way. Y'all will just very carefully so you don't bump these, but start a similar pattern here. There you are. All right, I got it.
Sandy,
Yes, sir.
correct me if I'm mistaken, but the over are in Iowa.
Yeah, they're no more is different.
Yeah.
Cliffs of Moher is different. Those are Ireland, and those are. I've been there. That's cool.
We were there with.
Yeah, you were there.
Right now, one of those lollipops.
Yeah, you have a lollipop. Here, I'm trying to get a big thing. All right, let me put these up on the counter where people can reach them.
Thank you.
All right, and that's it for today. How you doing, my friend? Yeah. Did you make that out of a? No, you didn't. Yeah. How the heck did you?
I got a bunch of paper. They're in like long strips.
That's wild.
So now I'm making a lot of stars.
Okay, I'm kind of blown away by that. It's so tiny.
Yeah.
Wow.